Good morning, students. Today we will continue the topic chemistry of carbohydrate, competition number 3.1, and today's subtopic is functions of monosaccharide. Here we are studying the functions of monosaccharide as a energy fuel and glycoside and its therapeutic importance. Functions of monosaccharides, we start with trioses. We have seen two examples of trioses, that is glyceraldehyde and dihydroxyacetone. So these two are present in the form of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, and they are intermediate of glycolysis. This glyceraldehyde 3-phosphate and dihydroxyacetone phosphate on reduction also forms glycerol, which is component of lipid. Next is tetroses. Example, erythrose and erythrulose. These are present in the form of erythrose 4-phosphate and erythrulose 4-phosphate. They are intermediates of one of the important pathway which is called as HMP shunt pathway. Coming to the functions of pentoses, first examples, ribose, ribulose, xylulose, etc. These are the examples of pentoses. These pentoses are also intermediate of HMP shunt pathway in their ester form, that means in the form of ribose 5-phosphate, ribulose 5-phosphate and xylulose 5-phosphate. Ribose sugar, all of us know, it is found in RNA. It is also found in ATP, NAD, NADP, etc. One of the inborn error of metabolism of pentose sugar is called as pentosuria. Inborn error means present since birth. There is a pathway called as uronic acid pathway. In this pathway, sometimes the enzyme xylitol dehydrogenase is absent. Due to deficiency of this xylitol dehydrogenase in uronic acid pathway, the pentose sugars are not further metabolized in that pathway, and hence the pathway stops at that point and pentose sugars are excreted into the urine, leading to a condition called as pentosuria. Hexoses. The important hexoses are glucose, galactose, fructose. All of us know that glucose is the main sugar found in the body. All tissues of the body utilizes glucose for energy purposes. Brain and RBC are totally dependent on glucose for their energy need. Another hexose is fructose, which is present in the seminal fluid, and sperms use this fructose for energy. Galactose is a component of lactose, which is a milk sugar found in the milk of lactating mothers. Coming to heptoses, example glucoheptose and pseudoheptulose. Pseudoheptulose is an intermediate of HMP shunt pathway. So these are the functions of various monosaccharides. Before moving further, we will see about an important test which is called as Benedict tests. This test was given by all sugars with free functional groups. Example, all monosaccharides, galactose and maltose give this test positive. The principle of this test is the sugar with free functional group forms enadiol, which is an unstable compound. This enadiol donates the electron, and this electron is added to the cupric ions present in the copper sulfate of Benedict's reagent. In the Benedict's reagent, the cupric ions are present, Therefore, the color of Benedict reagent is initially blue. When you add sugar to the Benedict's reagent, this sugar donate the electron to the cupric ion and convert the cupric ion into cuprous ion. This cuprous ion combines with OH group to form cuprous hydroxide, which has yellow color. After heating, this cuprous hydroxide undergoes dehydration to form cuprous oxide, which is red color. That means initially the color of Benedict's reagent is blue. After adding sugar and heating, it first get converted into yellow and then it becomes red. So this is called positive Benedict test, and it is given by the sugars which have free functional groups. Such sugars are called as reducing sugars. Now we will see in short about the various structures of monosaccharides. In the left, you are seeing the open chain structure of glucose. Here, glucose is called as D-glucose or dextrose. This is because OH group on the fifth carbon is present on the right side. Now, what is the importance of this fifth carbon? This fifth carbon is called as reference carbon in case of hexoses. When we number the carbon atoms in the monosaccharides, we have to go from above downwards. So, the last carbon of any monosaccharide is called as alcoholic carbon, and the carbon which is adjacent to the alcoholic carbon is called as reference carbon. In case of hexoses, the fifth carbon is the reference carbon. If OH group is present on the right side of the reference carbon, then it is called as D-glucose. So this is the open chain structure of D-glucose on left side. On the right side, it is a closed ring structure. And how this ring is formed? If you see, the first carbon of glucose, which is a functional group, combines with the reference carbon with the help of oxygen atom, and this forms a ring, and this ring contains 5 carbon and 1 oxygen atom. So it is a heterocyclic ring. During this ring formation, the functional group acquires a hydroxyl group. If this hydroxyl group is present on the right side of the functional group, it is called as alpha form, or if the hydroxyl group is present on the left side of the functional group, then it is called as beta form. So the complete name of this closed ring structure is alpha D glucose. So this type of structure is also called as Fischer's formula. There is one more way to represent the structure of monosaccharides. They are called as Haworth formula. In the Haworth formula, if you see, first carbon of glucose combines with the fifth carbon of glucose with the help of oxygen atom to form a ring which is called as pyranose ring, and the sixth carbon, which is alcoholic carbon, forms a branch on the fifth carbon. So the structure is like this. It is called as alpha D glucopyranose or beta D glucopyranose. Here the first carbon and fifth carbon combines with the help of oxygen atom, and sixth carbon forms a branch. Now in this case, the first carbon is the functional group. On the functional group, if OH is present on the lower side, it is called as alpha carbon in case of pyranose structure, and if this OH is present on the above the plane or on the upper side, then it is called as beta D glucopyranose. So in this way, the structures of monosaccharides are represented. In case of ketosis, the difference is here the functional group is present on second carbon atom. Therefore, when open chain is converted into closed chain structure, at that time the second carbon, which is the functional group, combines with the reference carbon. Here the reference carbon is fifth carbon in case of exoses, to form a closed ring joined by oxygen atom. So this ring includes 4 carbon and 1 oxygen. So it is also a heterocyclic ring, but with 4 carbon. In case of aldoses, there are 5 carbons and 1 oxygen atom. So this is the Fischer formula of ketone sugar. This is the Haworth formula for ketone sugar. If you see on the right side, second carbon, which is the functional group, combines with the fifth carbon, which is the reference carbon, with the help of oxygen, and 2 branches are there. One branch is at second carbon and another branch is at fifth carbon in the form of CH2OH group. Again, if OH group at the functional carbon, which is second in this case, is present below the plane, it is called as alpha D fructofuranose. If this OH group is present above the plane at second carbon atom, here it is called as beta D fructofuranose. So these are the various types of monosaccharide structures. After studying the monosaccharide, we study glycoside. These glycosides have some therapeutic importance. Therapeutic means they are used for treatment of various diseases. Now first we will see what is glycosides. Glycosides are formed when hydroxyl group of hemiacetal or hemiketal carbohydrate react with hydroxyl group of other non-carbohydrate. Glycosides are formed. So first I will explain what is hemiacetal and what is hemiketal carbohydrates. Hemiacetal carbohydrate means a carbohydrate with aldehyde group or aldoses. When converted in from open chain to closed ring structure, they form hemiacetal ring, which includes 5 carbon and 1 oxygen atom that we have already studied. Hemiketal means monosaccharides with ketone group. When get converted from open chain to closed ring structure, they form a ring which include 4 carbon and 1 oxygen atom. So this is called as hemiketal carbohydrates. Coming to the definition of glycosides, when the hydroxyl group of hemiacetal and hemiketal carbohydrate react with hydroxyl group of non-carbohydrate compound. These non-carbohydrate compounds may be methyl alcohol, phenol or glycerol. So this is the diagrammatic representation of the glycoside. On the left side I have drawn a glycerol, which is non-carbohydrate. They are also called as a glycol. On the right side I have drawn closed ring structure of glucose. So it is hemiacetal. It is called as glycon part. See, the hydroxyl group of glucose and the hydroxyl group of the functional group of glucose combining with the hydroxyl group of glycerol to form the glycosides. In these glycosides, the functional group, which is the first carbon in this case, involved in the linkage formation with glycerol. Therefore, glycosides does not give Benedict test positive and they are non-reducing sugars. Here the same is written, as the functional group of glucose is not free in glycosides, they are non-reducing sugars. Now coming to the therapeutic importance of glycosides. The most important glycoside is cardiac glycoside. In this cardiac glycoside, the examples are digoxin and digitoxin. These two cardiac glycosides contain steroid as an aglycon part which stimulate the muscle contraction or which increases the strength of contraction. This property of cardiac glycosides are very useful in the patients with heart failure, atrial fibrillation and atrial flutter. In these cases, the heart becomes weak. It cannot able to pump the blood in the required quantity. So, if such patients are treated with cardiac glycosides, the strength of the heart muscle increases and it will pump the required blood. Now, mechanism of action of cardiac glycosides. Cardiac glycosides inhibit the sodium potassium ATPase enzyme or pump. The function of this pump is it transfers free sodium from intracellular compartment to extracellular compartment. At the same time, it will transfer potassium from extracellular compartment to intracellular compartment. If this pump is inhibited, it leads to accumulation of sodium inside the cells. Therefore, there is one more pump or one more exchanger which is operating, that is sodium calcium exchanger. Now this sodium calcium exchanger normally transfers or exchange sodium with calcium. In this case, calcium from the cell comes out and the sodium from outside enter inside the cell. It is a normal case. But when this sodium potassium pump is inhibited and more calcium is accumulated inside the cell, therefore when sodium potassium pump is inhibited, more sodium is accumulated inside the cell and no more sodium will able to enter inside the cell because already the concentration of sodium inside the cell is more. So as the sodium does not enter inside the cell, the calcium doesn't comes out, and this calcium within the cell get stored in the sarcoplasmic reticulum, and whenever it is required for muscle contraction, it is released into the cytoplasm. As more calcium is available in the cell, the contraction of heart muscle increases. Next glycoside is oben. It is also a inhibitor of sodium potassium pump and blocks the active transport of sodium. It is not used clinically. Streptomycin is another glycoside which is an antibiotic which is used to kill the bacteria in the treatment of tuberculosis. Glucovalin is a cardiac glycoside which is used as a flavoring agent. And last cardiac glycoside is fluorescein. In this, the fluoretin is a glycon part and glucose is a glycon part. So importance is it causes renal damage. When it is consumed orally, it leads to renal damage. So this is about the functions of monosaccharides and therapeutic importance of glycosides.